Hi everyone, this is Matt. Welcome back to my channel. So, today I want to talk to you about how to structure your PhD thesis. And I've seen quite a few videos on this on YouTube, and I feel like people tend to make it overcomplicated, and it actually doesn't have to be super complex. And I'm going to break it down for you today. And essentially, it should make it much easier for you to think about your PhD thesis and what you need to include. So I'm gonna show you my own PhD thesis. So here it is. Here is the table of contents for my own PhD thesis. Um, and let's just look at it a bit more closely. So um, you have your title page, acknowledgements, abstract, table of contents, list of figures, abbreviations. And then finally you have your chapter one, which is your introduction chapter. And here in my case, this ended up being 29 pages long. I think there were 6 pages of figures. So essentially, it was 23 pages of text, which was about 7,000 words in total. So this is how long my introduction was. And a lot of people say, oh, in the introduction, you have to have your aims, your hypothesis, and all of that. And I want to tell you, I don't necessarily think this is needed. And the reason for that is science is not linear, research is not linear. I remember when I started my PhD thesis, and I had a title when I started, obviously, like, we all do. And throughout my PhD thesis, my title changed so many times. And I completely strayed from what I was supposed to work on to something completely different. And that is normal, what happens to everyone. And the point I'm trying to make is that you research something and you get to a point where you're not getting any more data, but you already, researched quite a bit on this particular thing, and you just reached a technical problem, right? And you need to try something else. And this is what often happens to us, especially during the first 2 years of your PhD, right? Most of the people keep trying lots of different things until they find this one thing that really works for them. That is, um, that is going to generate a lot of data for them. And then during their third, maybe fourth year as well, they really hammer down on this one particular branch of science, right? However, you've also done so much stuff before that, and you probably want to describe this as well. So this is why science tends to be more granular. Your research over the period of the 4 years is going to be more granular rather than linear. You will research something here, you will research something there, and then finally you will research here, and this will go, and you will generate much more data here. But you still have these pieces there that are not exactly connected, but it's still worth talking about them. Even if they're negative data, people should know about negative data. I often say negative data is as important as positive data because it saves other people time not to research something because there is no point, because it's not going to get them anywhere. So this is why it's so important. And this is why when you look here, I actually don't have aims, objectives, research questions in my introduction. So let's look at it a bit more closely. So I have like a little introduction here. So I'll talk about it more in a second. But this is the hook essentially. Um, and then I start quite broadly. I talk about transcription-induced DNA breaks. So this was mm, the broad topic for me. And then what my PhD really was about was these things called R loops or RNA DNA hybrids. And then you can see that everything else, essentially everything else here, is then about R loops. So I immediately went down into the topic that was absolutely key and relevant to my PhD thesis. And that's so important later for your PhD viva, because you don't want people to be asking you about lots of different things. They want you want your examiners to ask you questions about your field of expertise. In my case, this was R loops. And this is why here the majority of that introduction is R loops. And it's just that beginning here that is not about R loops. The rest is. Maybe the R loop is not in the name of the header, but it is about R loops. It was just this here that wasn't about R loops. Okay, so this is the introduction, and let's look at it closely now before I go any further. So let's jump into introduction. Come on, there you go. So it's only half a page here of text, and this was the hook. Okay, you can pause the video, you can read this yourself, but the point is that I'm trying to get the reader's attention. Look here, each of trillions and trillions of human cells in our bodies can experience about 49 DNA lesions or DNA mutations per minute. Okay, and each of these mutations could lead to cancer formation, cell death, or accelerated aging. Okay, so this is why my topic of expertise is so important, because in each of our cells in a human body you get up to 49 mutations, on average 49 mutations per minute. Each of these mutations could kill you, so they clearly they need to be repaired, they need to be fixed. And this is what I was really talking about, right? And here you can see now, as a result, human cells evolved elaborate DNA damage response pathways that recognize and repair different types of DNA lesions, DNA mutations. And now I have a little segue to the next section. Some of these DNA damage repair pathways are briefly outlined below. And I have a figure here. So in a figure, I basically show how these damaged DNA are repaired. And then I provide a little bit more text. And you see, this is only a page and a half, all of that stuff. And the absolute point here is that this is not the stuff that I'm particularly familiar with. It's useful and I will need to refer to it later, but I just need to know it in a sort of vague manner. I need to be aware that these processes exist and I need to know roughly how they work. This is what I had to know when I was doing my PhD. And this is why I have them here, but I have a very high level overview of them. And this was important because the overview was high level, then the examiners wouldn't ask me very specific questions about these things. That was the point. So let's go back into this, and now in the end of that section, I actually don't really have a segue to the next section. But look, the next section says transcription can be another potential source of DNA damage, and this is the link to the previous sections, this sentence. And now I talk about this general umbrella called transcription-induced DNA breaks, right? But I don't talk for too long about them, maybe 3/4 of a page. And then look, the last two sentences here, I'm going to highlight them, is what is absolutely key. So furthermore, transcription can cause genome instability, not only through promoting topoisomerase-linked DNA breaks, but also through formation of RNA-DNA hybrids or R loops, right? R loops is what I care about, and this is how I bring them into the light. As R loops are the subject of this thesis, this is what I highlight here, where formation and links to genome instability and human disease are described in detail in the next section. So what you can see here is that I started with a hook, okay? So I started with a hook and then I went a little bit more specific. I outlined all over DNA damage repair pathways, very high level overview. But I went a little bit deeper and I started talking about the DNA damage that is transcription induced by transcription. Okay, I went a little bit deeper, and then I said, actually, what my whole PhD thesis is about is these things called R loops, and this is where I went very specific. This is the rest of my pyramid. Hook, high-level overview of the field, and then you go specific into what actually matters. So, in terms of the volume, hook is very little. The overview, the high-level overview of the field is very little again. The part of then the specific part, the part where you talk about stuff that is actually relevant to your PhD thesis, that is high volume. Here you actually need to spend time talking about it. But it needs to be relevant. It needs to be absolutely 100% relevant to your PhD thesis. And it needs to be the stuff that you know inside out. Because when you're writing a PhD thesis, you need to think about your examiners reading this and asking you questions. This is sort of the end goal for you. It needs to be clear and concise. And you can't essentially leave yourself vulnerable to questions about research topics you're not familiar with. Okay? So that's why this is so important. So let's go back to this. So what do we have? Later, I talk about R loops. Okay, let's go back to the table of contents so we can see what I just spoke about. So we have the introduction, then we have so that was the hook, then we went a little bit more specific, and then we went all the way super specific. So let's jump to that the ending of that last paragraph here in the introduction. So this is that last sentence. I'm gonna highlight it again. That last sentence of my introduction. Since it doesn't matter what it is, dApps. Since dApps can stabilize the target substrates, it is plausible to hypothesize that they could ensure swift prevention and resolution of pathological R loops. What is so key here is that I am stating a hypothesis. In the very end of my introduction, I am stating a hypothesis. Look, I'm not giving them the aims, I'm not giving them objectives, I'm not doing any of that. I'm just giving them a hypothesis, something to think about, something that is actually the seed thought for my very first results chapter, right? And essentially, this was the seed thought for my PhD thesis, for what became my PhD research. But the key point is, this led me to my first chapter. The results from the first chapter led me to the next set of hypotheses, to the next aim and objectives, which became my next results chapter. And then again, because this is how science works, you ask one question, you will have one hypothesis, and you get some data, right? You get some answers, and these answers pose more questions, and you get more and more questions, right? Each time you answer your questions, you get more and more questions, right? Does this make sense? So it's like a domino effect. The more research you do, the more you realize, oh, I could do this and I could do that, and so on. So that's very important. And this is how, by the way I'm showing you how to do this here is this is how we write PhD thesis at the Russell Group universities in the UK. And the Russell Group universities are like the Ivy League of the UK, of the United Kingdom. So this is the golden standard of how we would do it in the UK. This is how we would write a PhD thesis. So let's go back to the table of contents. So we finished the introduction, and then after the introduction, I had materials and methods, and that's something I can talk about in more detail in another video. But I think that is an incredibly easy part of your PhD thesis to write. And then I have my first results chapter. Okay, so chapter 3 is a results chapter, chapter 4 is another results chapter, and so is chapter 5 and 6, and then I have general discussion. So let's focus on these chapters. Let's think about them. So this is my results chapter, and now you know that what led me to this results chapter was that seed hypothesis that I had in the very end of my introduction. Okay, but look, I have introduction here again in the beginning of this results chapter. Now I have aims and objectives. I have methodology. Only when I have the actual data, the results, and when I have discussion. So this is actually a paper on its own. And if you have written papers and if you want to submit your PhD thesis as a um, collection of papers, this is exactly how you would do it. You would follow everything I've done so far, and then you would just insert papers here as your chapters. So I have 3 results chapters. If you had 3 papers, you would make them 3 separate chapters. This is how it would work. So let's jump into that chapter 3 and let's break all of this more. So I have an introduction here, and this introduction is only a page long. But I essentially took the key information from the actual introduction chapter that I have, right? That is relevant to this results chapter, okay? So this is the key information from the introduction that is actually relevant to what I'm doing here. And then I have the aims and objectives. And I write, the aim of this chapter was to carry out a DAP siRNA screen to find a novel ALUP regulator, okay? And this stems back to that very hypothesis I had in the previous chapter. Sorry, in the introduction chapter. That is all it is. It's just a single aim that links back to what I had in the end of introduction. And then this aim is broken down into some objectives, which are the smaller, which are the endpoints essentially. Okay. So the aim is the overall big thing that you want to do, but you can split it into smaller pieces. And these are the smaller pieces. This is what I wanted to do. And then from there, I spoke about methodology because obviously to answer these objectives, I had to do some research. I had to do some experiments, and I decided what kind of experiments to do. And the point of that little section here, which is what again a page, maybe 3/4 of a page, is to talk about limitations. Is to talk about why I chose these methods, why these are good methods for what I'm planning to do, but also to talk about the limitations and highlight to the reviewers what is possible and what is not possible with these by using these methods, and maybe tell them why I haven't chosen any other methods. That's also very, very important. So, following this, I moved to my results. And now this is all my results data. And now from the results, I'm gonna jump, results, some figures, and so on, right? So this is the standard stuff you would expect. And then following the results, within this very chapter, I have a discussion section, okay? The discussion section doesn't have to be that long, so it's a page and a half so far, 2 pages and a half. Okay, another half a page. So 3 pages in total. So now this is absolutely important because you don't want to leave discussion for the very end of your PhD thesis because by then people will forget, the reviewers will forget the details of what they were reading about. This is why it's relevant to have this as a standalone discussion immediately following these results, to discuss them, to, you know, to link them to the literature, to talk about the limitations of these results, implications of these results, perhaps what should be done next, right? So this is why you have a discussion here to maintain a flow that is going to take you to the next chapter. Or it may not take you to the next chapter. You might actually say, okay, this is where we ended because we just got negative data, and because of that we decided to try something else. And this is how you actually maintain a flow from one chapter to another. So let's go back to the table of contents. So we have introduction, materials and methods again. There you go. This was one results chapter. Then this results chapter led me to the next results chapter, right? Which again had an introduction, which was relevant. But introduction was relevant to that chapter. And now I am actually talking about these proteins here called USP11 and synataxin. I haven't spoken about them in my original introduction. So this is new information, but overview of USP11 and overview of synataxin is new information, new introduction, but is key to this chapter. Okay? And then again, I have aims and objectives for this chapter, for this part of my research. I have my results and I have my discussion. And then again, I have another chapter, and look, now in the introduction, I talk about CRISPR-Cas9. This is stuff that I didn't talk about in the original introduction again, right? Imagine if I had to talk about all of this in my introduction in the beginning, I wouldn't be able to connect these things without explaining my data. I wouldn't be able to say, okay, I'm going to talk about ALUPs, then I'm going to talk about these two seemingly random proteins, and then I'm going to talk about CRISPR-Cas9. How are these things connected? I don't know how to explain this without revealing my data. It's not going to happen. So this is why I have it all here. And then again, aims and objectives for this chunk of my research, the results I've got, and then discussion, evaluation of these results. And then another results chapter, introduction, that contains overview of CAP1, which is another protein, right? So we have another seemingly random disconnected protein. But this is where the data from the previous chapter led me. It led me to this protein. So this is why now I discuss this here. This protein is only relevant to this chapter, to the data for this chapter. So I have aims and objectives, I have then results, and I have discussions. I evaluate the data again. And then finally, I have general discussion. Okay, so this is my last chapter, it's general discussion. And you can see this is not a particularly long chapter. Started on page 136, ended on page 143, so only 7 pages. But there is a general overview of everything I've got, okay, of all the data I've got, just a high level connection of everything. Then future directions, so what I would do next with all of this data. Suggestion that this protein called USP11 could be used as a drug target for novel therapies. So again, this falls under future directions, and it could be a subsection of future directions, but it was such an important part for me. I wanted to make this a heading in its own right. And then finally summary. And that seems pretty redundant, but look, that summary here is just like maybe 50 words, maybe 80 words altogether, just to summarize my 4 years of really, really hard work. But it's just it's just a touch in the end to say, so all in all, looking at all of the data and all of the discussion and future directions, um, future experiments that we should do. This is what I think altogether. So this is it. So if you um if you are going to write a PhD thesis using your papers, you would still have introduction just like I did. You would have then you could actually skip methods because you would then just insert your papers one after another, and then finally you would have your general discussion just like I did. So all in all, what I'm trying to tell you is that when you write your PhD thesis, remember work is not your research is not linear, and you don't have to make things up and really struggle to make it look like it was linear, like it was beautiful and you just moved from one experiment to another. It's just a straight line. Well, we know what it's like. It's like that. It's all over the place. Everything happens. And this is why you start with your introduction, and in the introduction, you talk specifically about what is, the most important to your research, and then, um, from there, you eventually get to that one hypothesis, that research gap, and that hypothesis is the the basis for the aims and objectives of your first research chapter, right? And then in this research chapter, you will have its own introduction with relevant data to that particular research, and then you will present your data, and then you will discuss your data. Because remember, PhD thesis. Well, I spoke about PhD thesis in another video, and I'm gonna link it in the description here. But the point is, your PhD thesis is full of information. Is just a sentence after a sentence is packed with information. And even if your PhD thesis is not long, and by not long I mean maybe like 120 pages, there is still so much information that you can't expect your examiners, your reviewers to remember everything. And this is why you need to keep them on their toes, and you need to bring them all the relevant information as you go. So this is why you need an introduction in every results chapter to just bring to their attention what is key for the set of data we're about to see. And then you need to discuss it straight away afterwards and tell them everything, evaluate that data immediately. Because if you leave that discussion, that evaluation for the very end, for the last chapter, then we will forget about this data in the first place. So this is why each results chapter is like a paper on its own. It's like a little document in its own right. Ideally, you should be able to just extract it out of your PhD thesis and be able to publish it on its own, right? It needs to be independent. So this is it. This is how we need to structure this. If you have any comments or questions, please leave me a link please leave me a comment in the description down below. And also I will leave a link to my other videos on how to write a literature review. That will definitely help you with writing that part of the introduction, because that part of the introduction is nothing else but a literature review. And also I will leave a link on my strategies for writing a PhD thesis. It's about the mindset. It's about how you should carry out conversations with your supervisor, how you should time it, and so on. So good luck, and if you benefit from this content, please subscribe, and I will see you in the next one.